Bismillahirrahmanirrahim. Rabbi shrah li sadri wa yassir li amri wahlul uqdatan min lisani yafqahu qawli. I am Dr. Mudassir Shabaz, Associate Professor, Sahara Medical College, Narowal. Previously, we have discussed about the development of kidneys in the chapter of urogenital system. Now, today we are going to discuss about the development of genital system. As we know already that the two systems developed from a urogenital ridge and is in the intermediate part of the mesoderm, and here we can see that here on the ventrolateral side developing the kidneys, and on the medial side developing the gonads. So from this urogenital ridge the urogenital system develops, and from one part the gonadal system develops. So regarding the development of gonads, first few things that should be understood. The gonads are basically two, the testes and the ovaries, and the both gonads, they are derived from three sources. One is the mesodermal part, as we have seen on the posterior abdominal wall, and other is the mesenchyme, the embryonic connective tissue. And there are primordial germ cells which will migrate into the developing gonads and produce the germ cells. And there is one stage initially, the two gonads, ovaries and testes, they cannot be differentiated from each other in the initial development, and at that stage is called as indifferent gonads. So it is very important to understand about the indifferent gonad stage, and it's on the fifth week. There is a thickened area on the mesothelium, that the mesoderm intermediate part, ventromedial part of the posterior abdominal wall, and on the medial side of the mesonephros that develops the indifferent gonads. Now, we see the origin of the and migration of the primordial germ cells. Occurs in the beginning of fourth week. The primordial germ cells, they appear along the endodermal cells in the wall of yolk sac close to the dorsal mesentery. There we can see of the hindgut. So the source of primordial germ cells is endodermal, and it is close to the wall of the yolk sac, close to the dorsal mesentery of hindgut. Here we can see this is the origin of the primordial germ cells, and from here the cells, they will migrate to the gonadal ridge, and this process completed fourth to sixth week. Now the primordial germ cells are actually, they are large spherical sac cells. In the fourth week they are endodermal in origin, and during the folding these cells, they are incorporated into the embryo, and thus the primordial germ cell migrate along the dorsal mesentery of the hindgut. So way of migration is along the dorsal mesentery of the hindgut. And in the sixth week, the primordial germ cells, they enter underlying mesenchyme and are incorporated into the gonadal cords. So here we know about the primordial germ cells. Now here again shown the part of the genital ridge, and we have come to know that this is the intermediate part on the posterior abdominal wall medial to the developing kidneys is the genital ridge. Now, sex determination, whether male or female, that sex determination is chromosomal and genetics. So there is a testis determining factor, TDF. The sex determined region of Y gene on the Y, which is sex determining region, and there is a gene which is called as SRY gene. So the absence of Y result in the ovary formation. So it is the presence of Y chromosome and SRY genes that will determine the differentiation of the sex of the baby. It's not the it's not the presence of double X, but it is the absence of Y chromosome. Although the genetic sex XS or XY is determined at the level of fertilization, but later development of the baby, of the fetus, of the embryo, whether it will develop into male or female, that depends on the presence of Y chromosome. The embryo's gender is not distinguishable for the first 6 weeks, as we have seen early, and this is known as the indefinite period of development. So here in the first 6 weeks, we cannot differentiate that this embryo is going to develop into male or female. So the characteristic for either of male or female genitalia can be recognized by week 12 of the development. So it's the 12th week that from the ultrasonography or other means we can determine the sex of the embryo. But up to the 6th week, it is indefinite period of development. So we cannot say where it will go. But we should keep it in mind that it is the SRY genes on the Y chromosome. It is the presence of Y chromosome that determine the sex of the embryo that will further show itself. Now, here we see the Y chromosome and here is the SRY gene component. So, in the 7th week of development, SRY genes on the Y chromosome activates a number of genes and gonads develop as testes. With no SRY gene, gonad develop as ovary. If there is SRY gene, that will activate other genes and will determine the sex. But if there is no SRY gene, so that will automatically develop into the female. Here again we see is the XX and here is the testis determining factor, which is responsible for the SRY genes, and that's the hormone that develop testes, and that then develop the hormones, testosterone and other hormones that will lead to the male formation. So here we see in 85% of the excess male have the Y-derived sequence. This is the abnormal development. If there is excess double X, no Y genes, but there is Y-derived sequences present, that will lead to the development of XX male. Single gene SRY with no conserved binding domain, SRY confirms male development in transgenic mice. 20% of XY females have deletion of point of maturation of SRY. So that is the congenital anomalies that can result from the presence or absence of SRY component. So SRY necessary but not sufficient for sex differentiate. Here in the males, in the XY females, 20% cases, SRY is present but not sufficient for the sex differentiation. Here again, another slide showing the genetic factors. So it's male development showing how the male develop, and here is the female development. And if it is going to be male development, so SRY through VTI1 factor and then the SOX9 SL1 to the AMH anti-Mullerian hormone is produced, and from here the star 3 beta HSD and other factors, they also influence on the anti-Mullerian production of the anti-Mullerian hormone, and then this sequence leads to the development of male. But if there is no, then the next one, that is the gene for the female development, and the testosterone is not produced, but it is converted to the estrogen, and that lead to female development. So here is the indifferent gonads. You can see here on the medial side of the developing kidneys is the genital ridge, and this here we cannot say whether it is going to develop into testes or ovaries. So in the indifferent stage of the uh, development of testes ovary passes in two stages. Number one is the indifferent stage, and it's before the seventh week or sixth week. A stage of differentiation between includes development of testes and development of the ovaries. Then a genital ridge and is a coelomic epithelium and primordial germ cells. Here, if we see in this slide, this is the genital ridge, and here is the coelomic epithelium, and here is the primordial germ cells which are coming along the dorsal mesentery to the developing gonads. So indifferent stage actually consists of three parts. There is a genital ridge, there is a thickening or differentiation of coelomic epithelium that will lead to the sertoli or follicular cells. In the males, sertoli cells, and in the females, that will lead to the follicular cells and primordial germ cells. So up to this level is the indifferent stage. Three components. So when we'll say indifferent stage is up to development of these three things. Here we see that the gonads are developing, but we cannot say whether they will develop into male or female. The epithelium is differentiating, coelomic epithelium. Inside are coming the cells, primordial germ cells, and the cords they are going to develop, but we cannot say they will develop into the follicular cells or the sertoli cells or sex cords in the male. So this is indifferent stage. In the ducts of the indifferent stage, we can see here the ducts, there are some ducts they are going to diminish, and other ducts they will develop. So here we see the duct along the uh, mesonephros, the mesonephric duct, that will become ultimately connected to the gonads. But if it is going to develop, all the ducts are present here. Paramesonephric duct, which leads to female development, and mesonephric duct is there, and the allantois is there, urogenital agea, developing a bladder. Here we cannot say that it will develop into male or female. So all the ducts are present. Now, from this stage, the definite gonads they are going to form. And first we will see how the testes they are going to be formed. Development of the testes. So embryo which contains Y chromosomes and the gene SRY genes on the Y chromosome, that will have a TDF, testes determining factor. The SRY is expressed in the mesenchyme and cause the cascade of events leading to the formation of internal male organs. The first step is tunica albuginea. Here is the surrounding coelomic epithelium that will thicken and it will form the tunica albuginea that will be converted into a thickened membrane, fibrous connective tissue, and that separates the cords from the surface epithelium, tunica albuginea. And within this, here are the seminiferous cords they are developing. And the mesenchyme here inside will develop into the leading cells. Leading cells here. Okay, other things that will diminish, that will not develop, such as the paramesonephric duct or other things. Here we see mesonephric duct is going to be connected to the developing gonads. So mesonephros itself will disappear, but the mesonephric duct will be connected to the developing gonads and will form the duct system of the testes. So what we have learned, that the surface epithelium that develops coelomic epithelium, that will thick and fibrous connective tissue, will give rise to tunica albuginea. Within this will be development of the seminiferous cords, and in these cords the primordial germ cell will enter and start developing between the mesenchyme, that will give rise to the leading cells that will develop the hormone testosterone. So primary sex cords, they starts proliferating from the coelomic epithelium and condensing extend to the medulla of the testes. In the medulla, the sex cord they branch anastomosed and form ratty testes. Here they will form the ratty testes. The prominent sex cord become the seminiferous or testicular cord. They lose their connection with the germinal epithelium with the development of tunica albuginea. Here is the tunica albuginea will develop. It will lose connection here. Inside will develop the seminiferous cords, where the primordial germ cell will come, and on here it will develop the it will branch, and here it will develop into ratty testes, which is then connected to the mesonephric duct through the ratty testes, and it is connected to the mesonephric duct. Now we see that is the testis determining factor. It induces the genital cords, seminiferous cord. They will condense and extend into the medulla of the indifferent gonads, which branches anastomose, and they form the ratty testes. A dense connective tissue, fibrous tunica element, separates the testicle from the surface epithelium. So in the fourth month, the testis cords become horseshoe shaped, and their activities are continuous with the ratty testes. And now, at this stage, they are composed of primordial germ cells, sustentacular cells of cytoli that are derived from the surface epithelium. So one thing here to note is that the leading cells, they are developing from the mesenchyme inside, but from the surface epithelium, the cells that are the sustentacular cells of cytoli, they will develop. So source of two cells is different. In the previous slide, if I go to the previous slide, it is a mesenchyme that give rise to the leading cells, mesenchyme here. But the surface epithelium will give rise to the cytoli, sustentacular cells of cytoli. So here, we see the interstitial cytoli cells. They are derived from the epithelium of mesonephric capsule and coelomic epithelium. The cells, they proliferate in the fetus, and mitosis ceases at the spermatogenesis. These fetal cytoli cells, they secrete the anti-Mullerian hormone that will cause the regression of paramesonephric duct. So we have come to know that AMH is produced, anti-Mullerian hormone is produced by the sustentacular cells of cytoli in the fetal life, and that produce AMH. Very, very important. Leading cells, they are developed from the mesodermal component of the gonadal ridge, proliferate and differentiate, that the mesenchyme. Here we see the tentacle side, and from the uh, leading cells, they here the leading cells, they are derived from the mesenchyme of the mesodermal part. And testicle guard formation proceed and is essential for the differentiation of the leading cell. These leading cells cytodifferentiate and secrete testosterone. So what we have come to know that these cytolysed cells, they produce the AMH anti-mullerian hormone, but the leading cells in the fetal life, they produce testosterone, which will lead to development of the external organs of the male development, external features of the male development, the development of the scrotum, development of the penis, and other. They will depend upon the testosterone. But non-development of the female organ, that is the paramesonephric duct, that will be inhibited by the cytoloid cells through the AMH. Here the AMH, which is produced by the cytolysed cells. So that's very important, and that can be included as the MCQ during the exam. The hormones which are produced by the two cells. So its overview here is that at the third week, the germ cell migrate from the yolk sac to the 4 to 6 weeks, these germ cells, they coalesce to form the primordial testis. Seventh week, cytolysed cells, they are appear, secrete the AMH, and they are developed from coelomic epithelium and differentiated, regulated by the Y chromosome. So, and then further development of the testes occur. So next, in the next thing to be done is the duct system, and we have seen that the mesonephric duct, which was connected to the ratis testis, here ratis testis of development of the testes, which are ratis. Here the ratis testis. Here is the mesonephric duct. Mesonephros is disappearing, but the mesonephric duct is connected to the gonads through the testes. So mesonephric duct persists, form the male genital duct, except the most cranial part, that the appendix epididymis is the most cranial part, epididymis of the appendix epididymis. And the mesonephric duct, it elongates, becomes convoluted, and form the duct of the epididymis. Here we see, here is the highly convoluted, and this is the appendix epididymis, but this here is the coiled epididymis, which is from the mesonephric duct. Then the further development, it becomes thick muscular coat, and it develops into the ductus deferens, which is then opens into the bladder through the ductules in the, say, we should say, in the uh, prostatic part of the urethra. They opens through the ejaculatory ducts into the prostatic part of the. So the region of duct beyond the seminal follicles is the ejaculatory ducts. These are formed by the mesonephric duct. The paramesonephric duct degenerate except for a small portion in the cranial and that cranial end in the that develop into the appendix testis. Do not confuse this appendix epididymis with the appendix testes. They are two different things. Okay. Then is genital glands. We will further discuss them in another chapter where the development of the glands. But the glands are the seminal glands and the prostatic gland. They are the two glands which are present on the posterior side of the bladder, and these produce there is a lateral outgrowth from the caudal end of each mesonephric duct, and that is the seminal vesicle, and multiple endodermal outgrowth from the prostatic part of urethra grows into surrounding mesenchyme differentiated into prostaticular glands. So two glands are formed, seminal vesicle which are the lateral outgrowth from the caudal end of the mesonephric duct. But the prostatic glands, they are prostatic part of the urethra, and that grows into the surrounding mesenchyme, and that differentiates the prostatic gland. Endodermal region. Bulbourethral gland developed from the paired outgrowth from the spongy part of the urethra. So these glands, they develop. So seminal glands, this we can see the lateral outgrowths from the caudal end of the each mesonephric duct. And the excretion of this pair of glands nourishes the sperm. And the prostate, multiple endodermal outgrowths from the prostatic part of the urethra and surrounding mesenchyme. Then is the external appearance of the genitalia first in the indifferent stage, then at the definite stage. So from here further on, so I will include in this lecture this thing. So external genitalia at the indifferent stage, here we can see the genital tubercle and the cloacal folds and the cloacal membrane here, and then further development there is the anal fold and the further development. So what we see in the indifferent stage is the third week. The mesenchymal cell originating in the region of primitive streak migrate around the cloaca membrane to make pair of cloacal fold. Then the cranial to the cloacal membrane, the fold unite form the genital tubercle. Caudally, the urethral folds anteriorly and the anal folds posteriorly. Another pair of elevation that becomes only on each side of the urethral fold, and then the elevations they appear. Later, these swellings form the scrotal swellings in the male and labial swelling in the male. This one is the scrotal swelling in the males and the labial swelling in the females, labia majora. So in the males, what happens under influence of the testosterone androgen produced by the leading cell? Rapid elongation of the genital tubercle, ALC, elongation of the genital tubercle, that form the phallus, during which the phallus pulls the urethra fold to forward, so they form the lateral wall of the urethral groove. Here you see lateral wall of the urethral groove. Phallus elongated and the urethral groove is forming. Then the urethral groove does not reach the most distal part. The urethral groove that does not reach the most distal part, so that is forming the, here you can see urethral plate and urethral pores. The epithelial lining of the groove, which originate in the endoderm, forms the urethral plate. So inside here, the epithelial lining that will form the, that originate from the endoderm, that form the urethral plate. So here, so this phallus part will give rise to the penis, and around which we will see in other chapters, we will see how the external male organs they are formed, or the penis is formed, and then we will see in the detail the characteristics. But here, for the purpose of introduction, I have mentioned these things so that they should be understood by us easily. So here is example of the hypospadiasis. They are shown that if there is abnormality in the opening of the urethra in the distal part and the external genitalia in the males, now it is forming, and it is the end of third month. The urethral fold, they cover the urethral plate forming the penile urethra, and then this canal does not extend to the tip of the phallus. The most distal urethral part is formed fourth month when the ectodermal cells they take part from the tip of the glands, penetrate inwards and form the epithelial cord. This cell obtain a lumen and forms the external urethral meatus. So external urethral meatus is formed by the ectodermal part. External urethral meatus is formed by the ectoderm. The genital swellings arising in the inguinal region moves caudally, and each one makes up half of the scrotum separated by a scrotal septum. So these swellings they will come closer together and they will further form the scrotal swelling which are united which in which septum they are separated from by a septum, the scrotal septum. Hypospadiasis, epispadiasis is genesis of external genitalia, bifid penis, and micropenis. They are the congenital anomalies associated with the development of the penis. That is the genital tubercle, actually. These are the anomalies associated with this. So, further we will discuss in the next is the descent of the testes, and that is an important one. And then we will discuss the development of inguinal canal and then the development of the ovary and other systems. So, inshallah, we will discuss that later on.